Welcome. I thought before we get started, today, so from an agenda perspective, I'm going to give a Global DARE Foundation update, and then Dr. Joe Hasia is going to give a research update. And we had planned for Dr. Radha Ramchandran to present on updated clinical guidelines, but I have not heard from her this week, so I don't know if something's occurred and she's not going to be able to present on that today. But we'll do a follow-up session via webinar, to cover that off, unless she does show, and then we'll be all good. Well, I did send her an email and give her the link and time, so hopefully she'll be able to come. If not, like I said, we'll do a follow-up via webinar. So before we get started, I thought I would play, I'm going to play a couple patient videos in between things. I thought that might be a nice way to kind of break up us talking and share a little bit about our patient stories and things of that nature. And so I'm going to play our We Dare to Believe video first. You would think that when you have a diagnosis, you would get a treatment plan and every doctor would know what to do for you, but that's not really the case with rare diseases.
I took my fourth DNA test a little over 5 years ago that gave me a diagnosis of Refsum disease.
I was night-blind at 18, so I became aware then I had a problem. I discovered Refsum when I was 50.
Prior to being diagnosed, I was, Ironman level triathlete. And then in 2017, I started to, have all of this left leg pain. And while I was riding and running, increasingly would have more and more pain. And then 6 months after that, I started to have this wavy distortion in my peripheral vision. It was really kind of scary time for me.
I now wear hearing aids. I use a service animal, a guide dog, to get around, and I no longer drive.
That proceeded on to seeing 15 different specialists and having countless diagnostic tests. I think at one point in time they actually thought I had MS, which wasn't correct. No one was really connecting the dots of what I had.
Nobody knew what was wrong with me, and then by a long series of lucky breaks, I was discovered by the one guy in England who knew what Refsum was.
Until recently, genetic testing has not been routine, even in the countries that have taken it farthest, like places like in Scandinavia. Norway's been one. Other countries have tended to be behind on this, and only now are these type of panels being introduced.
Refsum is a genetic disease that affects every cell in the body.
Caused by mutations in 2 genes that we know of. Patients are born normally, they develop normally, they often go through the early stages of life completely unaware of this disease.
It can cause loss of vision and blindness. It can cause neurological issues like seizures. It can have an impact on what somebody eats. It can have an impact on how a person is mobile and exercises and participates in physical activity.
So best way to diagnose this disorder is to check phytanic acid levels in blood, followed up by genetic testing that encompasses these 2 genes.
Usually people don't think to test for this disease. Usually there needs to be some sort of symptom present before a doctor decides that they're going to test for this particular disease because it is so rare.
The only therapy currently for Refsum disease is this low phytanic acid diet, which, phytanic acid isn't listed on any food label, so it makes it very difficult.
It's a healthy diet, basically no dairy produce, no red meat, only non-oily fish.
Before we started DARE, only 150 foods had actually been tested for their phytanic acid content. So we're often kind of guessing what is safe to eat and what's not. Not only do we have to worry about a low phytanic acid diet, we also have to worry about releasing any stored photanic acid because it accumulates in your fat cells, your nerves. Anytime you burn fat, like exercise, or you get sick, or say you have to go for a surgery and they tell you to fast. Well, all those things we have to be very careful on, because any release of stored photanic acid will actually increase your levels, increase symptoms. And when you lose your vision, you don't get it back. When you lose your hearing, you don't get it back.
To raise awareness for this disease, there's a lot of things that people can do. Being engaged with foundations like the Global Dare Foundation, they have a lot of opportunities for not just scientists in the field, not just patients in the field, but anybody to sort of put the message out on about Refsum, whether it be through social media channels or in their own local community.
The Dare community gives me the ability to talk to other people who also have Refsum. And with the rarity of Refsum disease, it's really nice to know that you're not alone.
It's important for anyone to remember that anything is possible. If anything that we've proved over the last 2 years in establishing Global Dare Foundation is we just need to come together as a community, whether that's patients, caregivers, researchers, clinicians, dietitians, all coming together towards that final finish line. I spent a lot of time racing in triathlons, and now I'm on a different race. And I want to get to that finish line of finding a cure. There we go. All right, so let me get started. I want to start the presentation a little bit, talking a little bit about the background on Refsum disease, what we've accomplished at Global Dare Foundation to date, and then what are our strategic objectives going forward. And talk also about some of the challenges that we continue to face and what do we have ahead on the next 12 months. So let's talk a little bit about Refsum disease. I think a good number of you in the room already know a little bit about Refsum disease. Either you suffer from it, deal with it on a regular basis, or you're a clinician or researcher that knows a little bit about it. But for the sake of those that are joining will watch this video later or attend online, I'll talk a little bit about Refsum. So it's a metabolic disorder. It's genetic. We've got 2 genes that we know of that are the primary cause of the disease, the PYHY and PEC7. And individuals with Refsum can't process a fatty acid called photanic acid, which, I had never heard of before this being diagnosed with this disease. And I said, well, what's a fatty acid? And, a fatty acid is what our body uses to use as fuel, as we do things like exercise and other activities. It's a long chain, branch chain fatty acid that's generally broken down or is broken down, in special compartments called peroxisomes. Because in Refsum disease we can't break that down, it accumulates in our fat cells and liver and tissue throughout the body. And as you can see, on this diagram, it impacts. It's a full body disorder. And so if you have high concentrations of photanic acid, you're going to have challenges, across many different functions in your body, vision, hearing, and if you have highest levels, you can have cardiac issues. So really important, the folks with Refsum disease, manage their levels of photanic acid. So a normal diet has maybe 60 milligrams of photanic acid, and we need to manage down to 10 milligrams. So the more we can avoid consuming photanic acid, that will help to minimize symptoms. And the sooner we can get people diagnosed, the better their prognosis is and quality of life, because as I talked earlier, accumulation makes a difference as well. So it's really important that we get word out to clinicians and individuals about the disease so people can be diagnosed earlier. Ultimately, we'd love to get people diagnosed at birth. Right now, there's not a mechanism for us to be able to do newborn screening. Genetics will really be the way to go in the future. So let me Move to the next slide. So just cover a little bit of what are the current therapies. A low photanic acid diet is one of them. Also managing metabolic stressors is another. So being, not that you can't exercise or you can't, obviously we're going to go through regular life. You're going to have stress, you're going to have illness, you're going to have those things. But how do we manage those things? Making sure that you have the carbohydrates you need to be able to not have that release or limit that release of photanic acid from stores. Another therapy is plasmapheresis. So if in general, plasmapheresis is used during acute management of the disease. So if people have really raised photanic acid levels where they're having significant neurologic symptoms or other things that are going on, they'll have plasmapheresis. And it's not just the level that makes a difference, right? It's also that accumulation of the symptoms to make sure there is some variability in the test itself. So you always want to make sure you're looking not just only at the level, but also the symptoms. There are some countries that use plasmapheresis on a more regular basis. There's mixed feelings on whether that's beneficial for patients. There hasn't really been enough research to know. And I know that's, as the team puts together clinical guidelines, that's the area they've struggled with the most on what is the guidance on when you use plasmapheresis. And the other piece is symptom management, right? There are definitely things that can be managed. As an example, someone can have something like macular edema. Well, that can be managed through eye drops. People could have cataracts. They can get cataract surgery. So there's certain things that can be done to improve quality of life by doing that symptom management. So a little bit about DARE. We have a simple mission. Our goal is to improve the quality of life of people with Refsum's disease. We've started Refsum Global DARE Foundation 3 years ago, or maybe 3.5 years ago. We've brought together patients from 18 different countries. I think we started at about 26 patients that we were connected with, and now we're up to 65, 70 patients and just growing month by month as we get the word out and as we continue to raise awareness for the disease. So we started out in 2019. I just, did whatever I could to pull together, enough board members to create a foundation. With 3 patients, 2 family members, we created Global DARE Foundation. But over the years, we know for long-term stability, we really need to have a more robust, mature board of directors that continue to grow this foundation from now until forever, right? We'll need this foundation to support the people with this disease. So how do we make sure that it can sustain over time? And so we've recruited quite a few people, not necessarily individuals who have the disease, but people who can bring good skill sets to the foundation to allow us to be able to raise awareness, to market, to be able to raise the funds necessary to do the things we want to do in the programs necessary. So right now we have a 12-person board, and we've brought, like I said, people from many different disciplines to the board to try to make sure that we've got a good, diverse thought to the program. And then, we knew right from the start we needed a team of experts to help us. We're not medical experts as a board, and so how do we bring the right medical team together, medical and research team together to help advise us on and work with us to set our goals and priorities around research, help to drive things like clinical guidelines. And so we established this board, and they're experts, across different paroxysmal disorders, vision, many different skill sets across the board. And so we're happy to have them helping us. As I say, we're looking to create clinical guidelines. We're looking to advance research. We're looking to advance different centers of excellence across the countries, and so they're helping advise us and really doing a great job at it. So we've got a couple of them here today that you'll hear Joe talk. He's our chair at our medical and scientific advisory board, and Florian used to be our chair the last 3 years, and so we're lucky. Anne and Dad are here today as well from our team. So really great support from that group. All right, so what's our strategic priorities? We as a board, when we first got started, we set some goals and we delivered on the goals, but we thought, back in 2022, we really needed to sit down and create a 3-year strategic plan. Where do we want to bring the foundation over the next 3 years, and making sure that everything that we do is tied to our mission. And so, our goals are, we've kind of grouped them into 3 different pillars, research and care. So really important to not only, raise awareness, but we want to be able to, drive the research for better therapies. And also just by, understanding and getting, patients the care that they need. We see often, people will get bad advice because there's not clinical guidelines, or their own country that there isn't like anyone that really knows anything about the disease. And so, how do we get that education out there? How do we get some published guidelines? So we're spending a lot of time on that over the next, have been and will be over the next couple of years. The other is raising awareness. I'm always saying if all we did was get patients diagnosed earlier, we would make a difference, right? If we could get people diagnosed at birth, what a tremendous difference it would make. We actually know someone who was diagnosed almost right at birth, and he doesn't have any symptoms. So we know the diet makes a difference, and so if we can get people diagnosed earlier, we'll improve quality of life. And then just bringing the community together. It's sometimes it can, Alan talked a little bit, it's nice to have the community because it's a 1 in a million, right? There's not many of us out there, right? Just being able to come together, shared experiences, understanding what might work for one person might work for another, but just being able to have that dialogue is really helpful. And then I talked a little bit about sustainability. We need to make sure we are always looking at how we can make sure the foundation can sustain for the long term. So it's not we're here for a short period and then gone. We want to make sure we're here and we sustain for the long term. All right, and I'm going to talk a little bit about what we've been able to accomplish over the last 3 years. And these are, I tried to choose some of the things that we're most proud of. Obviously we launched Global DARE Foundation in October of 2019. But, one of the first things we did is we need to understand more about what our patients are experiencing. And so we launched a patient registry. And that's the first ever patient registry released for Refsum disease. And I'll talk a little bit more about what we've seen in those results, but it does make a difference. I knew when I got diagnosed, I'm really much of a data person, and it seemed like the way to drive research was to start a patient registry. So we set that as a goal right up front. We also launched a country ambassador program. We know things can be a little bit different across different countries. So how do we actually get to understand what are some of the challenges? What are some of the issues that patient faces in different countries? I was just chatting a little earlier. In the U.S., we can get our photanic acid level done in a week. We get the results back. In other countries, it can take months to get your photanic acid level back. That's a huge challenge. How do you manage a disease where photanic acid is your only biomarker that you can understand whether you're doing a good job relative to managing metabolic stressors and managing, your diet. So how do we overcome some of those things? Plus, it's also nice when someone gets diagnosed in a particular country and there's someone in that country, they can actually talk to someone in their own language. And we have a German ambassador that we've connected with her only over the last year, and she's out connecting with every German patient that they've connected with over the years, and really making a difference in making the lives of those patients. So it's great having that program. We hope to continue to expand upon it. Right now we have 11 ambassadors across 8 countries. So hopefully we'll continue to grow that. We established a food testing program. I really felt that was low-hanging fruit, right? Only 150 foods have been tested. And, think about 150 foods, what the guidance, how many assumptions had to go into actually creating the dietary guidance many years ago. And there was many things that as an example, I always use the nut example, but they had tested 1 or 2 nuts and they were higher in phthalic acid, and so really all nuts were really excluded from the diet. Now we've actually tested a lot of nuts, and we're actually there's some that still are on the stop list, but there's a good number that we can eat, and that's a nice, healthy, protein-based snack that we can have. And it makes a difference. Now, there are some things that move from, the go list to a caution or a stop list. And we're still working through some challenges related to understanding some of the new nuances related to some of the measures relative to the testing. But I think to me, it's been a really beneficial program. We've gotten lots of great feedback from the community on that program. And then, each year, we want to be raising, we wanted to set a Refsum awareness, either day, week, month, just so that we could focus in on raising awareness. And so last year we had the first Refsum Awareness Week, and I thought it was a really successful week. Each year we're learning a little bit more on what we can do. This year we did you'll see a couple of the patient videos we created, and I think those are really impactful. We've been able to use those to help raise awareness in communities like retinitis pigmentosa community. We might be 1 in a million in the general population, but within the retinitis pigmentosa community, we're 1 in 300, approximately 1 in 300. And so that's, how, if we can focus in on that community, that will really make a difference in it being able to get people diagnosed earlier. Part of our strategic objectives is to improve treatment and care. And we want to be able to get more expertise, identified. There's definitely, around the world, there's definitely quite a few specialists treating people with Refsum today. How do we get all of them to collaborate, have a better understanding of the disease? How do we actually, we've got a really strong center of excellence in London. They have the largest cohort of patients. And now we've created a center in Boston, and I'll talk a little bit more about that later. But the more expertise, the more collaboration, the better we can do to, understand the disease and really get people better care when physicians and are collaborating across the community. I'm really excited. We're funding our first translational research grant at Johns Hopkins. Joe's going to talk a little bit about some of the work that's underway there, so I won't steal his thunder. I'll let him give you all the juicy details. But also, we've done a lot to raise money, and we're really thankful to the community and all of our supporters. We've raised in the 3 years, we've raised $265,000, and for a small organization like ourselves, it's not going to pay for gene therapy, but maybe with things like this pilot study that we're going to be funding, hopefully that will actually get us some data that will lead to larger grants and larger funding in the future. And so I think we can make some incremental progress and help mobilize things forward. I always think of like a snowball effect of being able to do more. So I'm going to talk a little bit more about a few of the things that you saw on the prior page. Creating the patient registry. Like I said, I'm very much a data person, and I knew data makes a difference in being able to understand and provide insights. And, rare disease is no different. After I was diagnosed, I really wanted to know, how do you spur on research in your disease? And everything was pointing to a patient registry. I even actually started to start a patient registry before I started a foundation. I'm like, how can I get one started? And they're like, Christy, that's not how it works. You got to start a foundation, or you got to latch onto a foundation. So I reached out to the ULF actually and said, hey, would you guys be willing to start a foundation? And they're like, well, we're not quite ready for that. I'm like, oh, I guess I got to start my own foundation so I can get a patient registry going and mobilize research. So that's exactly what we did. We've learned a lot. I actually thought, when you build a patient registry, everyone would just dive right in and put their data in. But it's taking a little bit to get folks, mobilized. Not everyone wants to have their data shared. Not everyone wants to be fully engaged. And I understand that. Everyone's got a little bit different perspectives on things. So we're continuing to try to recruit patients into into the registry so that we can continue to gain more insights. We just actually had a paper published, on our registry data, so I'm really excited about that. And I think that's like probably the first of many to come. As we grow that population, there'll be even more interest in that data. I talked a little bit about the food testing program, so I won't cover much more of that, other than the fact that we're continuing to test more food. So every year we're testing more foods, and we're going out to the community and asking them, what is interesting to test. We try to go as close to the base element, so that, that if we know, a particular ingredient is good, then you can look at a food label and understand that that's safe to eat. And so we've gradually started to increase the information into the diet. And we've created this traffic light system now, which I think makes it easier to understand. I think we've had some shifts in understanding about how does phytol fatty esters really, convert to phthalic acid. I think we need more research on that. We don't know really if it's a one-to-one. We are, as a conservative nature, we are treating it as a one-to-one, but is that really the case? And so we'll have to think about ways that we can figure out, how to really understand that. I do think, the food testing program was a big win for the community, and it was something we could do, right off the bat. I talked a little bit of the fact we were excited, Florian Eichler and myself, was it a year and a half ago, sat down and said, how do we increase the knowledge of Refsum's disease in the U.S.? How can we bring better care, to patients in the U.S.? We have this great center in London, but that doesn't really help folks in the U.S. And so him and I collaborated and brought different specialists together to create this multidisciplinary team, which to me, as a patient myself, it's really difficult, having to go to, I don't know, how many specialists do I see? A good half dozen specialists on a regular basis. And I have a full-time job. I run this foundation, to being able to take time off of work and be able to do those things. Now that we've set up this center, I can call one person. He makes all the appointments. I go in for 2 days and I get all my appointments done. So I don't have to be out of work for however many days, because it's a couple hours to Boston. Doesn't seem very far, but it does. I have to stay overnight, right? And how many days would I normally have to do it if it was spread throughout the year? So to me, it makes a tremendous difference. And so it benefits patients from that perspective. But also, as people come to that clinic, we increase the knowledge of the disease, right? Right now, people might be cared for in different places across the country, and those doctors are not collaborating with one another, really bringing the data together, really understanding what are I think we somewhat understand what all the symptoms are, but what if you started to see this consistent thing across patients? We were actually talking with the ophthalmologist, and they're like, we do think there's some differences with the eyes in Refsum than in other RP patients. So what is that difference? Maybe that difference actually could be something that could be taught to ophthalmologists to say, well, if you see this, maybe they have Refsum, So I think the more we can bring that data together, the more we can understand the disease, articulate what are the actual specific differences, for Refsum's disease, so that maybe it can be more easily diagnosed in the clinic, because right now I don't think it's easily diagnosed in the clinic. And then also just to improve care, right? If we want to, Joe will talk a little bit about some drug repurposing ideas. I think being able to have a cohort of patients. I think I said the London clinic has 20 plus patients. Well, if we had 20 some odd patients at the U.S. clinic as well, that's 40 patients overall. We could easily do maybe a clinical trial between the two centers, right, and bring that data together. It would make a tremendous difference. So I'm really excited for many reasons on bringing this center together. And one of the things I remember in setting up just even the patient appointments is that, the different specialists get together earlier in the week before the patient comes in, so that they're all talking to one another as well, because we're just one person. Even though they see each of us in their silos, they're still collaborating and talking with each other about everything that's going on as a as a patient as a whole. So lots of great benefits. I won't talk too much about the mouse models because Joe's going to talk about that, but I'm really excited. Just the fact that we're doing research and having a mouse model, to me, like, I feel that gives patients hope. That there is someone who's interested in the disease, someone who's wants to help find a cure. And so it's maybe the start. It's a long journey. It's not something we'll have overnight, but before we got Global Dare started, no one was really doing much in the way of Refsum's disease. There was really great research in the past and a lot of things that were done to understand the disease, but then kind of it died out. So we want to spark it back up and let's move it forward. All right. This is just the team at the Boston Center. As you can see, I said a multidisciplinary team. We've got all of the different specialists. Like I said, Florian's here today. And then we've got an ophthalmologist, a metabolic specialist. We've got a dietitian. We've got a physiatrist, a neuromuscular specialist, and an ENT. As necessary, we potentially could bring some other specialists as we understand the disease to this team. All right. So a little bit on the patient registry results. I thought I would give you some of the things that I think are of most interest. We talked a little bit about time to diagnosis. We knew that, just anecdotally, it takes people a long time to get diagnosed. But when we actually look at it in the data, it confirms it, right? It takes an average time of 16 years to get a diagnosis and upwards of 40. Could you imagine, like, you'll hear Sharon's story in a little bit, and she was diagnosed with retinitis pigmentosa at 11. She didn't get her Refsum diagnosis until she was 50. She has 5 degrees of vision. So time to diagnosis needs to change. So hopefully, as time goes on, we'll maybe see this metric come down if we're doing a good job. So it's interesting on average P, that chart in the middle that you see, I remember, asking our dietitians, like, well, what's a good level, for a patient with Refsum's disease to get to? And they're like, they really should get to 200 or less. But if you see on there's a good percentage of patients, I think almost 50%, that can't get their levels below 300, so what does that mean? Is it that they're, the results also say people are 96%, good to strict compliance with the diet. Well, what begs the question, like, why are their levels so high? What is it that's causing? Are there foods that we don't know that are high in vitanic acid? And we just don't know, and we think they're safe. Or are there other factors at play relative to metabolic stressors? Maybe they're not managing those as well. Or is 200 really achievable? Right? Like maybe that's not really the standard. Right? Like maybe there is different ranges for different people, and there is a safety level that you could achieve. And really, it's not 200 or less. So I think lots to learn there and dig in a little bit more. So prevalence of systems, it was interesting looking at this and seeing, 75% of the folks were diagnosed with genetic testing, but also like in the literature, it shows that about 30% of the patients have shortened fingers or toes, but in our survey, it's showing more like 2/3 of the folks. That's like a visible sign of the disease. Like if ophthalmologists just ask people to take off their shoes and socks, they might actually diagnose more. Now the patient might look at them like they have 2 heads, right? Because why are you asking me to take my shoes and socks off? But I think Bart Leroy actually on our medical and scientific advisory board, he says he asks all patients to take off their shoes and socks, because he is able. The bone abnormalities are present actually in several ophthalmologic conditions, so it's just interesting as you kind of look at the different metrics across the patient registry. We also wanted to understand, how are people being treated? Like, what are some of the regular tests they're taking? How often are they seeing specialists? So as part of it, it was also help inform our treatment guidelines. As you can see, from various things that some people don't get their levels done even only yearly, which I get mine done monthly, only because I'm very much a data person. I feel like it's the only really way to know I'm really managing things well. But it varies, relative to guidance and what people are asked to do. And there's some things that people from different types of tests and things that are done across the different patients. So this next, I thought I'd talk a little bit, about the journey for a patient as well. It's actually, when you don't have a diagnosis, it's very difficult, right? Like you're like, what is it? What is it? And then you get the diagnosis, you're like, oh, I'm going to get I think I said in my video, you're going to get a treatment plan, you're going to know exactly what to do. But that's not always the case, But then also you have to deal with the fact of, the emotional aspects of being diagnosed with a rare disease. You've got, guilt as one, like if I don't manage, my diet well, what if I go on a holiday or go on vacation? Can I have like a treat or do things like that? Like you feel guilty, right? Like, oh, am I gonna, affect my vision or am I gonna affect my hearing? You feel, constrained. Like some people have, physical disability related to the disease, and that can be a challenge in being able to send, like, how is that going to impact, work? Then there's also, young women too who want to become pregnant, or like, how am I going to manage that? And so, really trying to understand the constraints associated with that. Anxiety, being able to know, like, well, what if I get sick, or what if, things occur, like, are my levels going to spike, and what is that really going to mean for me? Similar from a fear perspective, and not knowing, if I have to, because of my vision, have to stop working or not able to, have some challenges related to being able to work full time, well, from a financial stability perspective, is that's an issue as well. And it's a burden, living with a rare disease. I mean, I was saying I often have to do lots of tests and go to lots of specialists. That's a challenge in a day-to-day life of having to, just do normal day-to-day stuff, right? And then I think that you get to a state too where you want to give back. You want to be able to, help the community, and you want to be able to raise awareness and do different things. Kind of like some of our country ambassadors are out there, helping with the individuals with the disease, or trying to advocate for raising awareness. So there's just a whole life cycle of things that we experience as we get diagnosed with a rare disease. Let's talk a little bit of some of the challenges. So, talked a little bit about long time to diagnosis. Like, we need to change that. We know that if we can get people diagnosed earlier, it's going to make a difference. Newborn screening doesn't exist, as I mentioned earlier, but are there things that can be done? Like, how can we, most people get that retinitis pigmentosa diagnosis. So how can we, get information out to the ophthalmologist? How can we get them to think about Refsum disease? I'd love it if everyone who got diagnosed with RP that didn't do a genetic panel, that they did a photonic acid level. Like, I mean, it's an inexpensive test, right? Like, and it would let us know whether someone is suspect for Refsum disease. So what can we do to educate and raise awareness? Lack of clinical guidelines, to me, is a real challenge. It's a challenge for a patient because they're not armed with what they can ask for, and I think it's a real disadvantage for specialists because they don't really know. And I think at times people are given the wrong advice. If someone goes for a surgery, the doctor doesn't necessarily understand that person should be treated differently than someone who doesn't have Refsum disease because they can't fast, right? So, you need to be able to eat or drink at least carbohydrates before you get into a surgery, and then you need a glucose IV through the surgery and until you can eat again. But a lot of times people aren't given that advice, and then they have significant losses of vision, significant other issues relative to their hearing or other neurologic issues. So it's really important that we get these clinical guidelines published and available, even if they're consensus guidelines, right? It doesn't necessarily have to be evidence-based guidelines at this point, but something that just, like I said, gives the specialists the things they need to know. Obviously, every patient is different, so it's not going to be a recipe for everyone, but it'll at least be the foundation that you can start from and then go from there. And then hopefully as we build these different centers of expertise, there are places they can also collaborate with. Like we actually have a dietitian network now where the dietitians are talking to one another across countries and trying to understand, well, what's working, what's not working. As we understand if we can't really understand how Vital fatty esters is converting, well, is anecdotally across patients, like how are things, as people limit things, are they actually seeing benefits and levels and things of that nature? We know there's not a lot understood about the accumulation of photanic acid. We know it accumulates in your fat cells and your liver and throughout the body, but we don't know actually what it does when it's there. Like, does it actually create any other harm? I know when I went for shoulder surgery, I actually had them take out the whatever was left of the tendon there. It sent it down. Ann Moser was nice enough to do a little testing on it, and it was full of photanic acid. Well, did it actually, was photanic acid, did it play a role in that deterioration? The deterioration that they had seen was more than they would have ever expected for someone that didn't have an actual fall or accident or something like that. Not that I wasn't physically active. People do get, shoulder surgeries for just normal reasons. But at my age and the amount of deterioration, they were surprised. So, does photanic acid play a role? The diet, is a good therapy, but people still continue to progress with vision loss and hearing loss and other symptoms, and so we do need better therapies, and that's why we're starting up research and also trying to, as I said, gather more data and understanding about the disease, so that we can identify, are there other things at play, besides photanic acid? We know that does play a role, but is there anything else that plays a role? So that we need more than just the diet, and I think I got one more slide. So what are we focused on over the next 12 months? So we've got Anne just completed her set of 40 plus foods. So we're excited for the dietitians to incorporate those new foods into the diet. It'll probably be we're hoping summertime. We're going to have an updated diet guide, and there is definitely some foods in there that the community is really excited to understand how much photanic acid and where it kind of falls into our traffic light system. We're advancing research studies, so Joe will talk a little bit about that in his presentation. And then we're really going to double down on raising awareness in the RP community. This actually established a relationship in the U.S. with the Foundation Fighting Blindness. I'm actually creating a Foundation Fighting Blindness chapter in Maine, and then connecting with all the chapter presidents across the U.S. To actually raise awareness in every state where there's a chapter. Actually go out, do presentations at their chapter meetings, really talk about, like I said, Refsum is approximately 1 in 300 in the RP community. That's a smaller haystack. We should be able to find more patients there. So really doubling down, really making more connections with blindness organizations across the world as well. Like our country ambassadors are really spending dedicated time. We had a community social, and we invited the Retina UK, and they came in and talked with our patients, and now we're doing more, with them as well. And so we're just, like I said, really pounding hard, on raising awareness there. We're also going to spend more time presenting and going to conferences. I'm excited to say we've got a booth at the Association of Research in Vision and Ophthalmology out in Seattle next May. So we'll have a booth for ARVO, and we're actually hoping we can get on the agenda for Retina International while we're out there and talk a little bit about Refsum disease. If we have some good information from the mouse model studies, maybe we'll, from a retinal perspective, we might be able to do some presentations there. But it'll be the start of being able to get out to some bigger conferences. That's where all the ophthalmologists go. So I'm just going to go around to every ophthalmologist. Do you know about Refsum? Do you know about Refsum? You should know about Refsum if you don't, and hopefully we'll be able to raise awareness to the place where people are getting more diagnosed in those ophthalmology visits. We want to expand our country ambassador program. The more outreach we can do in the various countries, they can help us, connect with those blindness organizations, but then they can also be a resource for patients. Because we do get, patients, I'm starting to translate in Turkey, and I'm translating, I spoke to someone from Iran, so it'd be nice to have folks who can speak their language versus doing Google Translate or other means. Even though I think technology is really coming up to speed, I think Microsoft Teams actually has the ability to translate on the fly if you get their premium version, which is kind of cool. So, and that kind of leads into my maturing global dare, like always looking for new ways for the organization to have the tools, the technology, be able to have the services we need as an organization to continue to build out our programs, build out what we have to be able to provide to the community. So I think that ends my presentation. And so I'll open it up to questions. So thank you. Small but mighty group in the crowd. Hey, Florian.
So I just want to have a shout out here for Christy, who's just done such an amazing job.
I think we got to give him a microphone.
No. Here. It's live.
So I just wanted to thank Christy and highlight, what an amazing achievement she has accomplished here. And the reason I want to actually do this is it has another motivation. When you're a small community, sometimes you can feel very isolated and alone. But in a small community, every voice carries extra weight. And I think the most important thing about coming together is hearing your questions, hearing your concerns, hearing anything that's on your mind, and knowing that voice carries so much more weight. And Christy is a perfect example of that. In this small community, she has made such a difference. And I think it's such an amazing, she's an amazing role model here. But every one of you has the potential to bring your voice in, and clearly everybody brings different talents, different perspectives, and those will be very different from Christie's, but they're all necessary. So I just want to sort of encourage everybody, to contribute and, in whatever shape or form brought you here. So there's no one single path, and just hopefully you can find your voice.
Florian's make me blush, Thanks, Florian, for your comments. Absolutely true. Like we've, as I talked about earlier, we've got, the country ambassador program, and they all bring a little bit different, energy, perspectives, keep me on my toes, of thinking about how different countries operate, and, what the different, cultural differences are as well. So yeah, I agree. It makes a difference to have everyone's voice heard, in whatever way they want it heard, right? Like sometimes it's a private message to me, right? Like they don't want to be public. That's fine, We do have a great Facebook group too, where people can ask all sorts of questions. But I agree, whatever active role they want to play or not active, like for sometimes it's, you don't want to be active either, for whatever reason, but, or maybe, like I said, you private message me and we chat about different things, Right, anyone else have any questions? Anything from is Susan still the only one on? So I'm pretty sure Susan doesn't have many questions for me. Susan's our vice president, so she's fully, oh, hey, Patrick.
If I speak loud enough, do you think?
No, it's got it for the video. We want it to, yeah.
So I just wanted to ask you, because I kind of work in this advocacy role a little bit too, what do you need from your community? What do you need help with?
Well, I think, for me, is really understanding the community needs. Like I want, often we've started to survey the community around what they want from us, right? Like what are the areas of focus that they'd like us to spend, and being able to get that input makes a big difference on, you saw me put up my strategic objectives for the next 3 years. I would hate to have those objectives not be aligned with what the community needs, right? Because all we're here to do is make the people with Refsum's disease lives better, and if we're doing something that isn't, aligned to that, like I want to know, so, and if there are things we should be doing more of or different, I want to know so that we can pivot, we can do different things. So I would say for patients to be vocal and tell us what they need. I mean, we're always looking for volunteers, but I know there's also, challenges from that perspective too. People have a lot going on in their lives, and so. But yeah, if they could tell me, I think the biggest thing is if they can tell us what they need. That would be it. Thanks, Patrick. Patrick's a great collaborator, relative to different patient organizations, collaborate with one another, and it makes a big difference. We're all doing the same thing. So I probably wouldn't have the patient videos that we have done this week if it wasn't for Patrick, because he shared with me a great tool that made my life a whole lot easier in assembling. Yeah, assembling those videos. So. And every hour I can save in life, is helpful in delivering more. So, so thanks, Patrick. All right, anyone else? All right, so I was going to, I think, let me just double check. I think I was going to go ahead and play.
My name is Sharon Burston and I live in Melbourne, Australia. I was diagnosed with Refsum last year in 2022 when I was 51 years old. I was diagnosed with RP 40 years ago when I was 11, and I had so many of the symptoms of Refsum already then, but clearly the people who diagnosed me with RP at the time weren't familiar with Refsum. I had already so many of the signs. I've got unusual hands and feet. I even had surgery on my feet when I was 11. The orthopaedic surgeon clearly didn't know about Refsum either, or he would have looked into it further. I have no sense of smell. I don't remember ever having a sense of smell. And I get some pins and needles in my hands and feet as well, although that's more recent. I have had some hearing loss probably for about 8 years now. I've been to several ENTs about that. They didn't make a connection either. So there's clearly a lack of knowledge in the community amongst the medical professionals of what Refsum is, because there were so many signs that could have been put together, and then I would have had my diagnosis way earlier. It was really difficult to get a diagnosis. I needed to do genetic testing and it was hard to get genetic testing done. So I eventually became part of a research study to get the genetic testing done, which gave me the diagnosis of Refsum. I really strongly believe that as soon as someone is diagnosed with RP, they should just automatically have a blood test to check their phytanic acid level. That would instantly alert to any issues with Refsum, and then it could be confirmed with a genetic test if need be. But it would be so easy to diagnose. And if I would have known sooner, there is so much I would have done differently about my life. I would never have fasted. I would never have done crazy diets to lose weight really quickly. I wouldn't have over-exercised. I would have eaten completely differently. I do all those things now, but I'm at a stage now where, I mean, it's never too late, but I've been left with only 5 degrees of peripheral vision now. When I was diagnosed, I had 120 degrees. Normal is 170 degrees. Like I said, I'm down to 5 degrees of peripheral vision, which is extraordinarily difficult to live with, as well as many other symptoms of RP like night blindness, etc. I think that it is really important that ophthalmologists are more aware of what Refsum disease is, because there is so much that can be done to slow down the progression of the vision loss, hearing loss, all the different things that can happen with Refsum if you're diagnosed early. There's an amazing food chart on the DARE website. You need to go to a metabolic doctor, you need to go to a dietitian, you need to become familiar with that food chart. Get in touch with the president of DARE. Christy will have a chat with you. She'll point you in the right direction if there are things you can do in your local area, or the support group on the Facebook group is also an amazing way to connect with people and, find yourself some new recipes. It's hard to do. I found it really hard to let go of eating the healthiest foods that I used to eat, like green vegetables. That was really upsetting for me to have to stop eating. Salmon, almonds, lots of things that I used to eat for health, I can't have anymore. I have found substitutes. Like I said, they're not as healthy as what I used to eat, but obviously they're better for me to not elevate my phytanic acid levels. And yeah, good luck with it. It's not an easy disease to live with, but there are ways to manage it.
It's a pleasure to be here. My name is Joe Hacia. I'm a geneticist at the Keck School of Medicine at the University of Southern California. And today I'm going to give an overview of some of the adult Refsum disease research. I am calling an audible on this one because I put together a talk that was slightly more technical in nature, but luckily my partner in crime here, Paul Watkins, and I have are going to be providing an overview of the journey of developing therapies for leukodystrophies like tomorrow. And so I took a few of those slides. So it's going to be towards a more general audience with a bigger picture perspective, which I think is appropriate. And I teach medical students quite a bit, so I actually love the big picture. To give you a little bit of an idea of my own personal journey, I began when I joined the University of Southern California faculty in the fall of 2000. I had an interest in phytanic acid biology, but from a very odd perspective. You could tell I'm an odd man to begin with. So I was interested in ecology and evolution. So I was interested in how different types of primates metabolize or break down phytanic acid, given all the different types of diets from all these different creatures, from chimps, gorillas, or orangutans, etc. So I was interested in phytanic acid biology, and when I was doing an internet search at the time, one of the biggest hits that came up was Hugo Mosier, who's Anne Mosier's husband. And I saw that they were experts in measuring phytanic acid, so I reached out to Hugo, and Anne and Hugo were very kind in around probably 2001 or 2002 to invite me to Baltimore. And I met a whole group of individuals who were interested in peroxisomal disorder. So peroxisome is the compartment in your cells that breaks down phytanic acid. And that's where I met Paul Watkins, who's become a dear friend and family member to me, as well as Anne. And we started working on those particular projects, and we got some funding for it. But it was several years later that Anne invited me to a United Leukodystrophy Foundation meeting, which focused in on a general type of peroxisome disorder called Zellweger spectrum disorder, which also has one of its components is phytanic acid metabolism defects, in addition to many others. And I started meeting many families that were affected by these diseases, and I met many of their, physicians, and I was very touched by the community. And I, made a promise to one family in particular that I would dedicate the rest of my research career to try to do translational research to try to improve the lives of, their kids and the future generations. So that's really where I come from. And today's my birthday, and I like to spend my birthday with family, and I consider Anne and Paul and Christy and maybe even John to be family, you know. So and all of the rest of you here today. So it's a particular joy to share this journey with you. So when we talk about adult Refsum disease overview, and we provide it here, let's see if I can pass the intelligence test. Yes, it should be the right arrow, and I think I'm hitting the right arrow, and it's not progressing. The down arrow, that's not progressing either, Christy. No, we actually need someone with some intelligence, and that's why Christy's coming to the mic over here. All right, just the down here?
Down here, yeah.
All right, good. I'm good at the ACs, Gs, and Ts, but you know, these things with arrows going everywhere, I have no clue. All right, so the objectives here today is to discuss some of the ongoing research to develop and characterize a novel mouse model of adult Refsum disease. We have some exciting unpublished data that actually was just generated in part by Ann Mosier. And we're going to discuss the early stages of planning some drug repurposing studies for adult Refsum disease. So as Christy already showed this particular slide, that adult Refsum disease is a rare metabolic leukodystrophy. You're all familiar with the manifestations of disease, which can affect the senses from vision, hearing, and smell. We also know there's peripheral neuropathy. And it is primarily caused by these loss of function variants in this Phi-H gene. That's the vast majority of the patients. And it's caused by an impaired catabolism or breakdown of a dietary branched chain fatty acid called phytanic acid. And I'll show you just some of the structures of that in a moment, just to kind of give you an idea of how this is obtained in a diet. So the accepted hypothesis, though, is that the accumulation of these toxic stores of phytanic acid in various tissues is causally responsible for disease. So for those who are, well, right now I'm hitting every one of the advanced keys, and like a chimpanzee, like which I'm used to working with, and I am not seeing any types of change. So thankfully we have an ex-triathlete in the audience who could come up here. Maybe you have to click it for. All right. So I think the thing is it had to be clicking. All right. So people can only obtain phytanic acid from dietary sources, and it really does come from in the biological food chain from chlorophyll. So phytanic acid is involved from the catabolism or breakdown of chlorophyll in ruminant animals. So these are going to be animals that have like a specialized digestive system, like a cow, where there's like a second stomach. And what they can do is they could take chlorophyll that they're obtaining from their diet, from just eating plant material, and convert it to a molecule called phytol. So what I'm showing here is this is chlorophyll. And this little side chain here, this little part hanging off or tail of that molecule, that's called phytol. If a human being were to ingest chlorophyll, it would just pass through your system. But in one of these animals with these second stomachs, they can actually convert it to a molecule called phytol. Fish can do this as well. And when anybody, if a human being ingests either phytol, you can convert that to phytanic acid. A cow can do that too. So when you're potentially eating a hamburger or like meat that's really laden with phytanic acid, it's just due to the fact that the second stomach in that cow, through the microbes that are living there, the bacteria, liberate this phytol, cut it off, and then it converts it directly into phytanic acid, and the phytanic acid is stored in the fat of the meat. Right? Now it's kind of interesting from a biological perspective that the cows themselves, even though they have a lot of phytanic acid in their fats, they're not developing Refsum disease. It's probably it's getting sequestered. It's not being in at high concentrations, probably in the tissues that matter, like the brain or the retina or the auditory nerves, etc. All right. So you can obtain phytanic acid from either the terrestrial food chain, which we think is just going to your local Burger King, or you could go and get it from also fish, because fish are going to be eating like plankton that are full of chlorophyll, and they're also doing a similar trick of converting it to phytanic acid. And so phytanic acid itself, though, provides no known health benefits to humans. If you get rid of phytanic acid in your diet, you probably can't avoid it entirely, but at ultra trace levels, you're going to be perfectly healthy. So there is a need, obviously, for humans to be able to metabolize or break down phytanic acid, because if you don't, then obviously you have the health manifestations of developing Refsum disease. So what I like to do, and this is from some of the slides that I have taken from Paul, is to try to discuss some of the research initiatives that are basically trying to help people with adult Refsum disease. So I work a lot with the National Institutes of Health, which is an agency in Bethesda, Maryland, and it's really the largest funder of biomedical research in the world, particularly in the United States. But they also have a campus within Bethesda, Maryland, where they have, tens of thousands of people who do biomedical research. In fact, Paul was working at NIH at one part of his training as well. So in this one particular center at NIH, they have many different centers. They have centers that are institutes that are involved in the nervous system, those that are involved in, heart, lung, and blood, et cetera. There's one called the National Center for Advancing Translational Sciences. And when we say something translational, what we're trying to do is to take basic research and try to make, products or that could actually help people, that would be able to be beneficial to your loved ones. That's what, that's what we're all about. So what I liked about this particular slide from this government website, NCATS, as they call that center, patient involvement, patients at the center. Everything we're doing is in mind. We're keeping the individual at the center. So we talk about basic research. We could talk about preclinical research, clinical research, clinical implementation, and then public health. So as you're going around clockwise, what you're seeing is becoming more and more practical towards actually creating products that can be used in the clinic. So I'm going to be focusing in on the basic research, which is going to involve scientific exploration to review, and it's going to reveal fundamental mechanisms, the biology, disease, or behavior. Like, for example, no one in our community knows exactly for sure why phytanic acid is toxic. We know that they accumulate in large stores in the tissues that are affected. But if you were to hold somebody's feet to the fire and say, can you tell me every step in the process, like a series of dominoes, we would not be able to, fully be able to explain it, all right? So there's still some gaps in the knowledge. And as a result of this, we would really like to understand that particular type of process, so that row of dominoes that's actually falling, that creates, retinal degeneration or is causing the peripheral neuropathy, so that we can intervene in a rational way, and we're not just randomly throwing energy at the problem. We're coming in a very directed approach. So animal models play a big role in this. So Paul and I will be talking about this tomorrow. But if you were to look across, biomedical research and go to the scientific session yesterday and in many other conferences, you'll see that there's genetically engineered mice, even fish called zebrafish or worms or flies. They're very frequently used in biomedical research. One of the reasons is that it's the accessibility to tissues. These animals are very small. They're very they're easy to keep up. They have about a 2-year lifespan. We could sacrifice the animals at any given point in time. We could be able to do invasive procedures so that we could be able to analyze their biology. It could be their brain, their retina, et cetera, other tissues, and be able to do studies that just can't be done in humans, so that we can learn something about the biology and also potentially test therapeutic interventions, all right? So it's not always guaranteed that the animal will develop health issues similar to the human condition, but we can make efforts to try to develop these animals that, reflect the aspects of adult Refsum disease. So from the great scientist Leonardo da Vinci, you might see he made the discovery that humans and mice, that they have 4 arms and 4 legs. I didn't realize. I always thought it was 2, but I see 4 over there. You can see the mouse themselves are a little bit more, they wear clothes as opposed to humans. So we're going to cover up certain human parts for any of the mice in the audience. But humans and mice, more seriously, have similar physiologies. And so because of that, we can be able to study these different organ systems, and we can be able to try to figure out why is vitanic acid so toxic to the retina, to the ears, and so on. And so there's many to dig a little bit into it, though. We just don't need sometimes one mouse model. We might actually have to do with many, many mouse studies, and I'll explain why. So that's partly because not all laboratory mice are the same. We can have different strains of laboratory mice, and I think of them as being like breeds of dogs. And you can hear my New Jersey accent of the dog, where I grew up very proudly many, many years ago. But the same genetic mutations, for example, we can genetically engineer. The technology's been around for decades to be able to disrupt a gene of interest. If it's Phi-H, we could create a mouse that really produces none of the Phi-H protein. The gene is completely inactivated. That mouse, in theory, which I'll describe, can't break down vitanic acid either. And so, but it turns out that just like there's many breeds of dogs, there's many breeds of mice. And sometimes you can take one breed of mice, or one strain of mice, I should say, and inactivate a gene, and the animal might be perfectly healthy. Another one, you can inactivate the gene, and the mouse has, a very severe disease. So as a scientist, it's like we are based many times just by observations and trial and error. So you have to go through many different approaches or many different mice to try to find the Goldilocks one, which will show the disease of interest or something similar to it that you can study. So what's really good about the adult Refsum disease research community is that we're building upon many decades of research. Even though it's been a very small research community, there's been fantastic progress, in my opinion, about, at least beginning to get the understandings of, what's the basis of the disease. It's vitanic acid accumulation. We have a little bit of knowledge about, why it's toxic, but we don't know the full story. But we started in 2008, our community generated the first mouse model for adult Refsum disease. When I say the community, it was done primarily in Amsterdam. One of the first author here, Sasha Fernandez, she's on the Medical and Scientific Advisory Board of the Global Dare Foundation. So we talk to her on a regular basis. We also talk to another one of the authors, one of the leaders in the field, Ron Wanders, who's around retirement age right now, but he's still very active in discussions about adult Refsum disease. So there's Sasha. This is a manuscript published in 2008. It's in the public domain. You could just look up Refsum and mouse on Google and probably be one of the first hits. So what they did was they generated a mouse which is homozygous for Phi-H null alleles. So when I talk about a null allele, that just means that there's no activity in that particular gene, right? So it's both copies completely inactivated. But they were on this mixed background, kind of like a mutt. They were on a background that is a Swiss mouse background, a 129S background, and an FVB background. So you can see now we start getting into the details of all the acronyms and etc. But you could just think of it as being three different types of dogs, three different types of mice. But they were when fed phytol enriched diets, not vitanic, but phytol, remember, is a precursor for vitanic. That can also jack up the vitanic acid levels in an animal. And it turns out when you do that, you will see, if I can be able to move, I can move this bar a little bit. I'm a little cautious over here. All right, so I think I semi-passed the intelligence test. All right, so in this particular case, the animals were fed a diet with where their chow was kind of soaked in or their food supply with phytol. And then you could be able to measure in various tissues, liver, kidneys, cerebellum, or brain, and testes, the relative levels of this vitanic acid. And they actually rise pretty dramatically. So control is a healthy animal that has 2 normal copies of that Phi-H gene. And the mutant animal, the ARD, that's the adult Refsum disease, that has 2 defective copies of that Phi-H gene. And so when you see this bar coming up, and this is at 6 weeks or at a 0.1% phytol diet, a little bit higher dose but a shorter period of time, you're seing that the relative levels of phytanic acid are dramatically rising in multiple tissues. So that's a good thing, because what it's showing you is that we could have an animal that we expose it to phytanic acid or a precursor, and the levels of this toxin are dramatically increasing in the animal. So that's step number one. You need to have the levels of the toxin increase, or there's no point to it, all right? So the problem, though, is that you'll see is like, well, they were on a diet 3 to 6 weeks. These animals do live a normal lifespan for a lab mouse is about 2 years. So when you're dealing with about 3 to 6 weeks or so, you're saying, well, that's a really short time frame. Why did they stop the experiment? Well, because the amount of this phytol or that they're putting in the diet is pretty toxic to the animals. So they're getting this decrease in body weight, like 25% in the males and about 16% in the females. And this could potentially be a little problematic because we're not seeing this in the patients. The animals are getting very sick pretty quick, but in a way that doesn't necessarily reflect what's happening in the patients. And in fact, this body weight loss was hypothesized to be due to real loss of white fat. So they became, extraordinarily thin in a way that would not necessarily reflect what's going on in people. So we have to be a little careful about that. In addition, what'll happen here, I think I'm going to try the magic of clicking the slide and then moving. Oh yeah, I can learn. Christy taught me. We looked at what that group did. I shouldn't say we. I wasn't involved in the study, but we look at this data quite a bit and we talk about it. Is that they also had abnormalities in their liver, which was a little different than what you see in humans. So if you were to take a cross-section of a liver from these animals, wild type is the healthy animals that are given various amounts of phytol in their diet. And it's stained to be able to pick up different types of the cells that are present in the liver. You'll see in the Phi-H null, this is the adult Refsum disease mouse, with higher levels of phytol, it looks like there's more of these white spots. And the white spots are like lipid droplets, like fat accumulation. They're getting this fatty liver. And it's not necessarily reflecting the disease that you actually see in people with adult Refsum disease. They're not getting this level of fatty liver. So you'd say, well, that's a little suspicious. They're losing weight and they're getting really fatty livers. Now, what's interesting to us is that in the cerebellum, which is going to be the part of the brain that's involved in controlling motor control, fine movement, there's a subset of neurons called Purkinje cells, and these are thought to potentially be compromised through their activity in some people with adult Refsum disease. And in diets, in the mice this is wild type mice, this shows you what it should sort of look like. I think it's this layer over here that we're going to be interested in. If you look in the null mice, there's a little bit less of these branchy structures here, which are going to be the Purkinje neurons. And so they're kind of missing. So the bottom line, I'm not a pathologist, but if you were looking at this with an expert, they'd say, well, there's certain of these cells are dying off. So that could be interesting to us because that potentially could reflect what's going on in disease. But at the same time, you do have an animal that is extraordinarily thin, not very healthy, and has some liver disease. And now there's some brain manifestations. There's something going on in their brain. But how do we know that's specific to to adult Refsum disease, or is just like general toxicity? So we really started. There was a team that was put together over at Johns Hopkins and the Kennedy Krieger Institute, which is basically nestled within the Johns Hopkins School of Medicine. That was all put together by Paul Watkins. So I do a lot of community building for different diseases, but in this case, this is really all Paul's doing. He masterminded it, and you should be very. I was pretty much amazed that we've been Paul, and Anne, who are now at the stage in their careers where they could be retired and they could be doing anything they really want. They are still involved in the research community. It's like very heartwarming to me. And what Paul did was we're continuing to talk about about some of the research I do, as well as my my personal life, but we'll leave that to another time. Paul reached out to a colleague by the name of Will Wong, who is in the Department of Physiology at the Hopkins School of Medicine, and he got him interested in phytanic acid biology and Refsum disease. And what Will did was take some of his excess research money, he doesn't have a lot of it, but he had a little bit that he could invest, and he says, oh, let's like start up a new project. And he started breeding mice that also had the Phi-H mutations, but they were on a pure genetic background. Instead of being part Rottweiler, Dachshund, and, you know, Pekingese dog or something, it's like pure Rottweiler. I say that because I grew up with a Rottweiler named Lucy, so I always think of her. So a pure background, that reduces variation. So we're, you know, that's actually a very, very good thing. And then, you know, as he started developing that model, one of Paul also was able to rope in one of his colleagues, a pediatric neurologist by the name of Joey Scafidi, who's also a proud native of New Jersey, over at the Kennedy Krieger Institute. And they started expanding that colony, meaning they made more and more of the mice. So there's a whole group of mice that are now that contain that harbor that the Phi-H null alleles, or basically are not making the phytanol-CoA hydroxylase, and they're happily breeding over at the Kennedy Krieger Institute. Now, as part of the team here, as Anne is always the quarterback, who's doing all the phytanic acid measurements and lipidomic profiling. We also have Waydad, who's one of. I am so glad that Waydad is on the team. She's been a dear friend for many years. I work with a physician scientist by the name of Nancy Braverman on other paroxysmal disorders, and they're over in McGill. And Waydad I've known for many years because she was a trainee in Nancy's lab. And a few years ago, I was talking with Anne, and we said, well, we really should have try to have somebody to take over some of these projects and, you know, in the future. And so we went to a couple of different foundations. So we went to Global DARE, we went to the United Leukodystrophy Foundation, the Global Foundation for Paroxysmal Disorders, and the RhizoKids Foundation. We said, can we collectively get some grant money so we can be able to fund somebody to work with Anne on these types of projects for several years? And we got the consortium of four foundations together, and there is Waydad here very proudly. As to me, this is when a scheme comes together. It's very, very rare that it ever comes together, but that was a dream, and she really is a dream. So you should talk to her and her family, and you'll be inspired. So the idea here was to develop the mouse model so we can develop, you know, the human phenotypes or human manifestations of disease without all this unwanted toxicity, you know, without the fatty liver disease, without, you know, any excess, you know, abnormal extreme abnormal exposure to phytanic acid or phytol, just something that reflects in a mousy world what's going on in the human condition. All right. So that way we could test disease mechanisms and potentially test therapies. And I'm more in the therapy testing mode. I mean, I love the basic science, but ultimately it's the patient first. And we want to just do research that, in my opinion, is going to most benefit the patient, hopefully in the short run. So this is brand new spanking data from Anne. And at the Kennedy Krieger Institute, they took the mice that they developed, and they fed it either phytol, that precursor to phytanic acid, or just phytanic acid themselves. And they looked at the amount of phytanic acid in their blood plasma. And what you will see here, this is at 0 weeks, and then this is at 3 weeks. This is kind of like baseline, and you're seeing over time at different types of diets, we're getting dramatic increases in the amount of phytanic acid in the plasma. So that is really, really good news, because now we have, you know, I have an animal in a pure genetic background that's going to be very reproducible that is showing this dramatic increase in phytanic acid levels. But even better is the fact that I've heard anecdotally, and maybe Anne or Paul can chime in now or later, that these animals are growing in, you know, at a normal rate. They're not losing weight, but they have the high phytanic acid. So that means probably all that non-specific toxicity is probably, you know, like that caused, you know, all the weight loss and loss of fat. That's just not happening. Now, it's still so very early on, we don't know, are these animals going to, over time, lose their vision, have some sort of retinal degeneration? We have no clue yet. We're going to be looking for that. We'll be looking to, measure, their neurological health to see if they can, are there problems with their movement or not. But, the first step is the plan is coming together. So, and that's basically all under our control. Everything that's under control is being done. We can't, predict whether or not the animals will lose their vision. If they do, it would be great for us, bad for the mouse, but great for us, because then we can potentially test some, targeted interventions and say, okay, we'll give the animal drug X. We'll see whether or not that improves their vision, slows vision loss, or potentially improves their balance. Right? So that's where we're going. And that was supported through this newer studies now through a Global Dare Foundation grant, and that's ongoing. So we're very excited about that. And then what we plan to do with that would be to use that data to apply to government grants so that we can get much larger sums of money to be able to really try to bring this home. So the drug development pipeline is a very long process, though. This is something that Paul will speak about tomorrow morning. But you could go from basic research to discovery, developmental research, and marketing. And if you're looking for something, like akin to, a cancer drug, it might cost you hundreds of millions of dollars. So when you're also noticing that in this particular diagram, when you're looking at the numbers of volunteers, well, clearly, I believe my understanding is that we have less than 100 patients in the registry for adult Refsum. So it's impossible to have over 500 volunteers. And so when you're dealing with the ultra rare diseases, you have to, think a little bit differently from the development of drugs for, common diseases. And obviously, although I'm sure John is going to be doing some fundraising, I don't think he's going to get the $460 million overnight. I might be able to cut you a check for $460, but I better get a dinner on that. All right. So it could cost it could take it could take decades and millions of dollars to develop these types of drugs. So what we want to do is not leave you depressed, but give you an idea about how the community thinks about this. Well, first, that's developing drugs. There's a lot of different other types of therapies that are out there. Now, for leukodystrophies in general, but it's not going to be applicable to adult Refsum disease. You might hear about hematopoietic stem cell transplants or bone marrow transplants. That's used for the treatment or prevention of the advancement of the cerebral form of adrenal leukodystrophy in children. There's other types of technologies for different types of diseases where you can replace the proteins themselves or enzymes. There's genetic therapies, and I'm very involved in the gene therapy world. Many of my colleagues are as well. This whole meeting is full of people involved in gene therapy. So that's something to think about. But it still is a little bit further down the road. Even the least expensive gene therapy trials that I've known from various groups in ultra rare diseases, it might cost somewhere about $7 to $10 million for a small clinical trial. And I've worked with organizations that have, raised that amount of money and had their children treated for one disease is called giant axonal neuropathy. But it's a caution that even though they were at the forefront of having their children treated, but to date they haven't received the benefits that they had hoped for. So you always have to try to keep this in mind. So one way around this, again, try to keep a more happier perspective on this, because I think in adult Refsum disease, in my opinion, of all the disorders that I've been involved in working on, this is the one that is the most eminently treatable, or at least have interventions that can improve somebody's quality of life. We are just really dealing with trying to lower the amount of a toxin in a person's body and try to make sure that it's not getting to the tissues or organs of interest. So one way around trying to get around these hundreds of millions of dollars, even tens of millions of dollars trial, is to use drug repurposing. So there is, the idea here is that there's an unmet need, that some people with adult Refsum disease cannot control their circulating phytanic acids despite being on the Refsum diet. And this is a good portion of the population. And I've learned that from just working with Global Dare. There's a difference for me as a basic scientist about reading the literature and reading papers that are decades old and actually meeting people and asking what's going on in your life. So I am blessed when I have that opportunity, either in person or over Zoom, to talk with the folks that you're trying to help. So as a result, if you can't really control this vitanic acid levels that are circulating, the thought would be you'd be at increased risk of accelerating vision and hearing loss in this peripheral neuropathy. So at this point, I did a brainstorming session with our adult Refsum disease, our global DARE, I should say, medical and scientific advisory board. And because it's brainstorming, rule number one, do no harm. I'm not recommending any of these ideas for the treatment of any patients, but just to show what's going through our mind and with some hope that, some of them we could potentially, go forward and try to implement in clinical trials in the future if warranted. So and this really comes from being a basic scientist who does a lot of medical teaching, usually year 1, year 2 medical students. And occasionally they're going to be taught, they're invariably going to be taught pharmacology very early on in their training. And there's this concept called pharmacokinetics. And the four tenets of pharmacokinetics has to do with absorption, distribution, metabolism, and excretion. So you can just look at it as that vitanic acid is a toxin. You want to either not be exposed to it, or you just want to get it out of your body as fast as possible, or you basically just want to keep it away from the tissues where it's really, really toxic. So absorption, that's the process by bringing the substance in through the diet. Distribution, how it's spread through the body. Is it in the fat? Is it in the retina? Is it in the heart, brain, etc.? Metabolism, how is it broken down into, in this case, we talk about alpha oxidation for vitanic acid, how it's broken down at 1 carbon and 2 carbons at a time. And then excretion, how do you get it eliminated from the body? So the practical ways of regulating it, well, the mainstay is the diet, and that's been pretty remarkable what Global DARE has done, funding these types of studies to be able to figure out, like the vitanic acid content in diets, and that's all been, led by Ann Mosier. And then there's also, and so in this case, you could think about, for your Harry Potter fans, this would be the vitanic feasts and how to avoid them. So that's one, and then so this is like the mainstay of the treatment or prevention. Then there's orlistat, this intestinal lipase inhibitor that some people take that has some unpleasant side effects, but it's basically going to prevent reduce the absorption of vitanic acid from that you ingest in your diet. It can just kind of pass through your system. All right. So some people have thought about that and even tried it, but at the same time, there's some unpleasant side effects of that. So the other way of going about this, going down the line from the tenets of pharmacokinetics, is distribution. Keeping it in safe harbors, where I think of adipose or liver, it probably would be far better to have your vitanic acid that's in your body in those places as opposed to your retina or your brain or your nerves, right? So that's what we want to try to do. And so I want to think about, is there any way of keeping the vitanic acid, in those places and not distributed elsewhere in the body? So we've been talking about drugs to be able to reduce the release of vitanic acid from the fat stores in your body, all right? So when you exercise, you burn fat, and then we want to try to find a way that we can potentially, in theory, if you could be able to prevent the vitanic acid or lessen the amount of vitanic acid that's released from your adipose and put into circulation, and it could wreak havoc elsewhere, that could be beneficial. And it's also known that vitanic acid is carried on particles called low-density lipoproteins. You might know LDLs from the field of the world of cardiovascular health, where you want to lower the bad cholesterol, LDL cholesterol. Well, these vitanic acids also carried on these types of particles like LDLs. So potentially we could try to find drugs that exist that might lower that. Now, the other way is increasing alternative ways of breaking down phytanic acid. So the mainstay of this of phytanic acid metabolism is something called alpha oxidation. That's the process by which you're chewing back the carbon atoms and breaking down the phytanic acid to simpler molecules. But when you have a mutation or a mutation in this Phy-H gene, you basically impair that main pathway. But there's a backdoor pathway. It's far less efficient, but if we can be able to increase that less efficient one, well, then you could be able to break down the phytanic acid, and that would actually be pretty cool as well. So this is an idea that's been around for many, many years. And many of the advisory board members, have been thinking about this. And there's some thought that there are some drugs called fibrates, which are very well, known to be used for the treatment for lowering, cholesterol, and for people, for many thousands of individuals take fibrates, and this could potentially have a secondary effect of increasing this omega oxidation, which is this backup pathway. So that's something that, we're discussing, within our community. Would it be worthwhile to potentially have a trial where we'd have individuals who have with an adult Refsum disease who have very high levels, still maintain high levels of phytanic acid? If they were to take some of these fibrate drugs, would it potentially reduce those levels in circulation? And that's something that, these are pretty safe drugs, very widely prescribed. So that's just something we are at least discussing. We're not necessarily going to move ahead on it, but something we're thinking about. And then eliminating it. As Christy Array talked about plasmapheresis, it's often used by some people in the community, but there's drawbacks to it, a little controversial at times. You could also use, a drug like orlistat, which could regulate the absorption and elimination of phytanic acid, but still there's many drawbacks to that as well. Christy came up with this idea that I always liked about sweat. Is there any way you can be able to eliminate some of this phytanic acid through perspiration? That's just something that is, we don't know it's still in the most basic idea now, because we don't even know how much phytanic acid is actually liberated in perspiration. But that's something you could probably, study in a person, obviously, or even in the mouse model. And then coming from California, I think as on every other sidewalk you might see in the Boston area, you'll see a Dunkin' Donuts. Over in Los Angeles, you'll see a liposuction clinic. So it would be possible, at least in my opinion, as an engineer, I would be thinking, if I have this toxin in my body and there is stores of it, maybe perhaps that might be one way of eliminating it. So you can see we try to think both inside and outside the box. So the questions to consider for any approach is, the risk-benefit analysis. Clearly, do no harm is in the forefront of our minds. So we'd want to, have any of these interventions, we'd have to make sure is appropriately safe, so that any risk that you're taking is far outweighed by the potential benefits. And then what's the minimal required preclinical information that needs to be gathered prior to testing in people? Do we have to test it in the mouse first? Perhaps we could, if it's a very safe drug that's very widely used, we might be able to do a small-scale pilot in people if they're willing. And what are the rate-limiting steps in putting together these small-scale clinical trials? They're not for free. They clearly have a lot of infrastructure involved. So that's something where Global Dare is going to potentially play a major role in trying to overcome these types of hurdles. So at this point, I'd like to quote another major philosopher here, Walt Disney. I only hope that we never lose sight of one thing, that it was all started by a mouse, and also supported by funding from the Global Dare Foundation. So thank you.
Any questions for Joe? Oh, let me get you a microphone.
So one question I have is, in research, has it shown any difference, I guess, in, say, a 15-year-old that's got the Refsum disease and their phytanic acids versus that same 15-year-old at 40 years old? trying to keep their levels down if they stay on the same diet?
It's actually a good question. I think, someone else may be best suited for this, Christy. I mean, I know that Ann has had many years of, following some of the patients, but obviously Florian as well, and I think Paul would know.
I think probably the answer is we don't know, right? Would you say, Florian? I mean, I think that's the reason we want the patient registry. That's why we want to start, getting basically, there's quite a few diseases that have natural history studies. They either do a retrospective, prospective studies that can kind of help us understand, because there are patients in our community that have been diagnosed earlier and then now are older. And so what has been their progression? What has been their course? But I think we were talking earlier, everyone's seems to be a little bit different too, even in the same families. Someone has a much higher level and they are very compliant, watch their metabolic stressors, and then there's another person in the family that doesn't do any of it and their levels are lower, and the other person's higher. So I think there's just so many factors at play, and there's so much unknown. I don't know if you want to say anything, Florian, or if that, I pretty much covered it.
Yeah, I think we're still at very early days to fully understanding what the future lies in the relationships between metabolism and injury, and as Christy says, I think it very much has to do not just with environmental factors, but maybe there are other genetic modifiers, and really getting a large enough cohort to understand this is critical. And we've worked with other diseases such as adrenoleukodystrophy for many decades, and still we're struggling with some of the questions around how to optimize diet and whether just to follow levels of certain lipids is good enough, and I think we're moving more towards the view that you have to look at many different things and understand what's happening in different compartments. If you're correcting this by diet, is it really getting to the compartment of interest? I mean, if you're measuring something in blood, is that, telling you what's happening in the eye or in the nervous system? Probably not. But together, I think we're learning a lot, and hopefully we'll have good answers for the young man.
So this is probably something that I should know, maybe. But you mentioned earlier about the phytanic acid in a cow. I don't still fully understand the whole where the phytanic acid comes from. So we talk about cows and we talk about the lamb, and then we got the diets as far as the pork is kind of back and forth. I don't understand exactly. You mentioned also like the diet of the cow, maybe.
Yeah, all of this originates from chlorophyll.
Mm-hmm.
And certain animals can be able to break down chlorophyll into a precursor of phytanic acid called phytol. And that phytol immediately gets turned to phytanic. But humans, no matter how much chlorophyll you ingest, and there's been long-term studies, I know Paul and I have talked about this, where there's persons eating massive amounts of spinach, just like laden with chlorophyll, and then you measure the chlorophyll, it basically just passes through your system. But in some animals, they break down chlorophyll and it turns into phytanic. And then you eat their meat, and then as a result of this, you're ingesting the phytanic acid. So your body isn't generating the phytanic acid from chlorophyll, but it's coming from an animal that can. Or the other alternative, which gets a little bit more complicated, is that precursor phytol is now being found to be present in some forms in certain types of plant materials. And so that's why when you're doing these dietary studies now, they're saying, oh, well, we not only have to look for phytanic acid, but then we have to look at the phytol content of food, right? So that's what's going on in the food chain.
So,
and these are good questions.
So depending on what the diet of the animal, could that depend on whether that animal could be eaten again? Like say the cow, depending on the diet or what is fed?
That's a good question. No, in a practical sense, cows are going to be in the food supply are going to be fed where they're going to be fed. They're going to be grazing.
Sure.
and depending probably on the breed of the animal and then the type of plant materials that they're ingesting, it might vacillate, go up and down. But at the same time, I think it'd be inevitable that virtually any source of beef is going to be high in phytanic.
I have corralled our other members of our Medical and Scientific Advisory Board who are here today, just to introduce themselves, so you can get to know them a little bit more, or how they, what's their background, how do they become involved in Refsum's disease, why it's of interest to them. I thought that might be good. But before we do that, I'm going to show another patient video, and then we'll bring them all up.
I live in Switzerland, but I was born and raised in Canada, and I left Canada at the age of 23. I'm now 60, and I have been living with Refsum since the age of 34. I think my Refsum story is a bit atypical in that my journey to a diagnosis was relatively short, thankfully, and it wasn't until the age of 32 when I started to develop issues with my peripheral vision. I was on vacation at the time, actually, vacationing in Ireland, and I started to get a glimmer on the periphery of my vision. And that led me to go to see the ophthalmologist. And at the same time, I had also lost my sense of smell. So I had gone to see the ENT specialist. And although I had told both of these specialists that I had these concomitant issues going on, neither of them really made any connection between sense of smell and the RP. When I got my RP diagnosis, the consultant nurse there gave me a pamphlet about a self-help group for people with retinal diseases by the name of Retina Suisse. I made contact with them very quickly because I wanted to find out more about the disease and connect with others who had been diagnosed with retinitis pigmentosa. I became a member of Retina Suisse, and I actually joined the board of directors there. I was a board member for 10 years. And actually, thanks to that, I got my Refsum diagnosis relatively quickly, because through a chance discussion with the president of Retina Suisse, I mentioned to her that I had also lost my sense of smell. And she clicked immediately, and she said that she's aware of a syndrome that presents with both of these conditions, and that although it's a long shot, I should be tested for Refsum. So I went to the University of Zurich and they did a phytanic acid blood test, and sure enough, my levels came back elevated. They were about 190 micromoles at the time. And because the diet sheet was so limited, it took me quite a while to actually come to terms with changing my diet. Thankfully, my Refsum journey has been, or let's say the progression of the disease has been relatively slow. My main problem is tunnel vision. So my vision has decreased to about 25 degrees. I was forced to give up driving in 2014. Thankfully, I live in Switzerland, which has an extremely good public transport network. I do have some loss of hearing, although I don't use hearing aids, certain voices that give me issues. And I do have recurring signs of neuropathy. They come and go. I know that despite following the diet, for some reason I haven't been able to keep my levels under control. 5 years ago, I reduced to working part-time, work at an international pharmaceutical company as a global trial leader. Thankfully, my company is one of those companies very advanced with diversity and inclusion. And working part-time with a disability is very much welcome there. A lot of people have various medical diagnoses. There's people who have just a hearing problem, or people who just have RP, or just have ataxia. But with Refsum, we have it all. The other thing that I find a bit frustrating with Refsum is that it's a deteriorating condition. So we're always getting used to a new normal. And I often think with my vision, okay, I've lost so much. If it would only stay like this, I can cope. But, there's this nagging thought in the back of my head that says, well, maybe it's going to get worse. How are you going to cope then? We don't, you just do your best to cope. But it's, so much going on at once that sometimes it's overwhelming. But generally speaking, I don't find it difficult to comply with the diet. Having said that, there are certain things that I miss. I'm living in Switzerland. This is the land of milk and cheese. That's one of the things that I really miss. Nothing tastes the same as a true sticky gooey mozzarella cheese on a pizza. Let's face it. No alternative will be that good. My symptoms are not obvious when somebody meets me initially. Because I use the cane more often, I am now telling people in advance, I do have this disability. And of course, doing all this awareness work through the DARE Foundation, of course, I identify as a person having Refsum, and I'm more comfortable about, owning it and raising awareness. When I think about improving quality of life, my main wish would be, to have a cure for Refsum. And I do believe that we will eventually have a cure. It may not be, in my lifetime, or I may not be able to benefit from it. The research that is moving along with the support of DARE, I think these are exciting times. But yeah, my one wish would be, to be healthy again.
Just briefly, I'm Florin Eichler. I'm a neurologist at Massachusetts General Hospital in Boston. And I came into the field of rare diseases really by virtue of having wonderful mentors in Anne and Hugo Moser. And I had the good fortune of learning from them not just neurogenetics and biochemistry, but how to navigate a field of rare diseases where there's so much uncertainty and so much unmet need, and how you pay attention to opportunities and serendipity, just the kind of thing that you just heard from Susan. I went to a retinal specialist who paid attention to the fact that my smell was off, and put things together, and paying attention to details is, I think, what Hugo Moser taught me, or I at least tried to follow his guidance and how he connected different fields. I after working at Kennedy Krieger, I then embarked on advancing gene therapy for adrenoleukodystrophy, which is another lipid disorder affecting the brain. And here the scenario was that bone marrow transplantation was the only modality that was able to stop a very devastating form of this disease. But it came at a price. It came at a price of engraftment problems, graft versus host disease, and a whole host of complications. And so we devised a new way of addressing this by using the boy's own cells. And this became the first ex vivo gene therapy that is now the first FDA-approved gene therapy for leukodystrophy. So this was about a 12-year path, but I think as Joe Hessia pointed out, these things are happening faster, and there's much more interest now in advancing gene therapy and potential cure for many of these rare diseases. What I think of day to day when I see patients is, among all the many treatment options, what is the right path to follow for this individual? What does the risk stratification look like? As Joe said, risk-benefit ratio is everything. If somebody is doing well with a metabolic problem, maybe don't want to subject them to a high-risk treatment that could have bad side effects. But if you are having some symptoms and you're seeing them getting worse, you might be more willing to step in and take on certain risks, knowing that there might be others that can guide you. There's medical establishment that could help manage side effects. So I think we are in a good position now compared to 10 years ago that we are starting to have options for rare diseases such as adult Refsum. And the thing I also learned from Hugo is to always ask for help. And I certainly need a lot of help. And I'm in a good place at Mass General where there's so many other specialists that we can bring together for adult Refsum, and bringing together specialists for the eye, for the peripheral nerve, for hearing, for metabolism and diet, has been a really nice scenario, and I think the Boston Center of Excellence is really striving to provide best service to patients in need, and also understanding, what kinds of guidance we can build up over time. As Christy said, this is probably the main thing that we need, but we can't just draw it out of a hat. We have to do this together. We have to learn together by sharing experience, by gathering more data, making more diagnoses, and understanding how all this data informs patients of tomorrow. Is that okay? Good.
Yeah, that's great, Florian. I don't know. Can you hear me? Ooh, yeah. So Paul, would you like to go next?
So, hello everybody. My name's Paul Watkins, and I'm kind of fairly recently retired from the Kennedy Krieger Institute after 35 years there. Florian mentioned serendipity. We also refer to that as dumb luck, but that's kind of how I got into this also. When I was a graduate student, I was working on fatty acid metabolism, not with respect to any particular disease or anything. But then I went off to to NIH to do postdoctoral research, which had nothing to do with fatty acids or anything else that's relevant to the rare diseases. But down the hall, I subsequently learned, were researchers who were like pioneers in Refsum disease, basic science research, and phytanic acid metabolism in the 60s. And then, so there have been kind of, I think it's fair to say there have been about 3 waves of basic science research into phytanic acid and Refsum disease. The first wave was in the 60s, where a physician scientist named Dan Steinberg started trying to figure out how Refsum disease was and phytanic acid were related. His lab was the one that fed some poor volunteer 2 kilos of boiled spinach to see if they would raise their phytanic acid levels, and it didn't go up. And then, so that was kind of the first wave. And they got the research on how phytanic acid is broken down and what the problem is in adult Refsum disease. They kind of figured some of it out. But then there was a lag period where not much was really happening to advance the field. And then when I was offered when Hugo Moser offered me a position at Kennedy Krieger, it was mainly to work on adrenoleukodystrophy. But then Anne introduced me to the Zellweger syndrome and the other peroxisomal diseases. And then somewhere along the line, we got interested in Refsum disease. And so a colleague, Stephanie Mihalik, and I started working in the lab in the 90s, so early mid-90s, on Refsum disease, and mainly trying to figure out how more detailed information on how phytanic acid is broken down and what the defect might be in the disease, because at that time we didn't know what the enzymes were, what the genes were, and so we were very lucky to be involved in the identification of Phy-H as the enzyme that's missing in Refsum disease. And then unfortunately, like things happen, the funding runs out and you move on to other things that are important, but other diseases. And then the third wave came with Joe Hasia and his renewed interest in phytanic acid metabolism in the non-human primates, which is how we met. And this eventually got continued on, and then when Christy came on, then it really got started again in earnest. And so I am pure basic science. I am, interested in the metabolism of fatty acids and mouse models, and how we might use them to advance this wonderful work that Christy has breathed new life into, essentially. And so I'm happy to participate in any way I can, and I'm happy that the mouse is eating the phytanic acid diet and not getting sick yet. We want them to get sick, but we want them to get sick on our terms. We want them to get sick with with the problems caused by the excess phytanic acid, so we can understand that better. We don't want them to be sick because of side effects that are unrelated to the disease. So that's where we are. Thanks.
Thanks, Paul. Any questions for, I didn't ask any questions for Florian either, but maybe we'll take questions at the end. But I do have to thank Paul because there's a lot there's a lot I don't understand about all of the biology. I don't think that was my strongest class in school, but he was I talked to him and I said, can you give me a breakdown of what is phytanic acid and like how does it all go through the system? And he gave me a little 101 when I first got started, so I really have to thank Paul for always answering all my why questions. I really appreciate it. So Anne, do you wanna go next?
So, good morning, everyone. My name is Ann Moser, and although I'm fully retired, I'm still working at the Kennedy Krieger Institute. There is so much to be done, and so I've continued the research. My first encounter with phytanic acid was in the late 60s. When my husband Hugo asked me to set up the assay of phytanic acid in plasma, and we were in Boston at the time. Hugo knew of Dr. Steinberg's work, and he realized that if we identified a patient who could not metabolize phytanic acid, that the condition would be treatable with special diet. So he reasoned that we should develop this plasma phytanic acid assay. So we did. And we received samples from an astute ophthalmologist at the Mass Eye and Ear Infirmary in Boston. And his name was Dr. Elliot Burson. And we analyzed over 300 samples, completely normal levels. These were all patients with retinitis pigmentosa. And then the 301st sample was highly positive. So we identified our first case of adult Refsum's disorder in a 30-year-old. And so Hugo started this man on a special diet, which was low in phytanic acid. And all these special diets had been published by Dr. Steinberg and the group in Norway. And the group in Norway is where Dr. Refsum was. And he had a group of patients as well. So then in 1976, we moved to Baltimore. And the patient with adult Refsum disease was from Philadelphia. So he followed us. So he came to Johns Hopkins for his therapy. And his blood levels were very stubborn because he had a lot of phytanic acid on board in his stored fat. So in order to help reduce that stored fat level of phytanic acid, he went to plasmapheresis at Johns Hopkins, I think about once a month. And he took the train from Philadelphia to Johns Hopkins for this procedure. And we collected the plasma that was removed from this patient, and we still have liters of it stored in a lab for studies, if anybody can think of a good study. So his levels gradually became more normal, and at a certain time, I would think over a period of 2 years, they finally decided that he no longer needed plasmapheresis because his levels were maintained with low phytanic acid diet. And about that time, this patient also took a trip to Norway with his wife and met Dr. Refsum. And I think while he was in Norway, they also made some suggestions for his diet. So this individual was a remarkable man, and he continued working until retirement age, but he also volunteered in his community. So he was a very giving individual. So his levels maintained at the same level for years, and but his retinitis pigmentosa progressed. So we last heard from this individual about 3 years ago, and then his wife called me recently to say that he had passed away at the age of 92. So he had lived a long life. We continue our interest in treating diseases, paroxysmal diseases, rare paroxysmal diseases, and there are other genes, defects where phytanic acid is impaired. Phytanic acid oxidation is impaired. And those include many of the PEX gene defects. And it also includes this disorder, rhizomelic chondrodysplasia punctata, where Dr. Falata has worked on this disorder. She has worked on the mouse model for this disorder. So I'm extremely pleased that Widad Falata has joined our lab. Her expertise is welcome, and she is a delight to have in the laboratory. So thank you very much, and I'll answer any questions.
Thanks, Anne. I appreciate it. Anne's modest. From a global DARE perspective, we're very thankful to Anne, because she really, we wouldn't have our food testing program if it wasn't for Anne. She's worked, extraordinary amount for us, and at a discounted price, I'm pretty sure. And so we've had some great enhancements to the diet that we would not have if it wasn't for Anne. So thank you, Anne. And we'll have Widad come up, and then we'll see if anyone has had any questions for our representative MSAB members.
Okay, hello everyone. I'm very happy to be here, first of all. My name is Widat Fallata. I'm coming from far away, so I was born and raised in Saudi Arabia. I did medical school there, and then I got interested in genetic diseases because also we had a lot of these genetic diseases in my country, and I remember I was going as an intern to these clinics just to see and connect with patients, and I was always get in touch with them, and then my heart broken when they said no treatment for now, and I was always looking for hope for those patients and family. So that's how I got interested in research. Then I got, thank God I was so grateful. I got a scholarship to study more abroad. So I did master in clinical research in genetic and dysmorphology, and that was in San Diego, California, in which we measured eyelashes in some of genetic disease, and I see that they have a longer eyelashes. And then I was further interested to get more in research, and that's when I started applying for a PhD, and I was very grateful to be accepted to be supervised as a PhD student under the supervision of Dr. Nancy Braverman, and that's when I got involved in paroxysmal disorders. So I got in touch with the phytanic acid when I started working with one of the paroxysmal disorders called RCDP, rhizomelic chondrodysplasia punctata, and I worked with patients and a mouse model. In this disease, which is there's a defect in PIX7, but those also patient and the mouse model, they have a high phytanic acid. And that's when we start measuring the phytanic acid for them. And then we're sending, I remember we're sending some bunch of the samples to Anne to measure phytanic acid for us. And we see that's increase in phytanic acid, and that's how we got interested also. And then after I graduated, I met so many fascinating people during my study and PhD, during going to the meetings and patients. So I promised myself to be committed to paroxysmal disorders, and I get involved in this. So I'm very humble and grateful to continue the work with Anne for paroxysmal disorders. So that's in summary. And recently, I was connected with Christy, and to be a member for the board for this community, and I was very happy to be one of the members. And I also want to thank Joe because he's a dear friend, and my supervisor, Dr. Nancy, because she is the one involved me and let me go with her for all the family meetings with the patients with paroxysmal disorders. And in those meetings, I remember, so one of the very first meeting, I met Anne, and I said to her, one of my dreams to be working with you, and here we go. So dream come true. So thank you, everyone, and happy to meet everyone here.
Thanks, Widad. It's great to have you on board and interested in Refsum disease. I'm always excited to have more people coming to the field, right? I think, and we're very happy that you can now begin to learn and gain all of the knowledge from Anne, right? We want to make sure that she has all the support she needs as well, too. She's an amazing soul. So, all right. Any questions for our medical and scientific advisor board that we can answer? All right. Well, I want to thank everyone for coming and engaging, and thank you, Joe, and our medical and scientific advisor board for being here and presenting today. So, thank you.